Continuing chapter 9. The caption is When Potato Salad Goes Bad. Potato salad's holding a gun on the condiments. They got their hands up. In sealed containers, a lot of what makes food go bad is anaerobic organisms because you sealed it up and there's not a lot of oxygen in there. But now, we're going to talk about aerobic respiration. Caption here says brain aerobics. I expect you can probably read the balloon. At some point, 1970s, I think it was, somebody decided that that the way to exercise was to exercise kind of continually at a level where you could just keep going and it didn't make you breathe really hard. And because if you work out hard enough that you lose your breath, then you're actually going into anaerobic phases of aerobic respiration. We'll talk about that. And they decided, okay, we'll do aerobic exercise. So, and it's not entirely accurate for what's going on, but you'll hear people talk about doing aerobic versus anaerobic exercise. Is it tennis is aerobic, sprinting is anaerobic, and jogging is aerobic. It's not a great definition. So, aerobic respiration arose when there was oxygen to be used. Once photosynthesis arose and oxygen became both built up in the organisms that were doing photosynthesis, they needed at the very least to keep it from being poisonous to them, because remember, oxygen's really reactive, it can be poisonous. But also, it turns out if you can put it to work, you can get it to do very wonderful things. And we will talk about that later in the chapter. Now, most of this you don't really need to know the details of. But you start with glucose. You break it apart out in the cytoplasm of the cell. This dates back to the original anaerobic respiration that the cells could do. And remember, mitochondria were kidnap victims? Is that the endosymbiont theory says that they were picked up and then put to use rather than being digested? Well, they're where the actual aerobic stages happen. So you break glucose in half a little bit, you get a little bit of energy from that, a little bit of usable molecules, produce a molecule that goes into the mitochondrion, and that's where the oxygen gets used. And here's the oxygen going in. It picks up hydrogens. There's a whole bunch of details here you don't need to know. But you've got glycolysis. You break the glucose in half roughly. You've got a cycle where you start to break the rest of it up and you get carbon dioxide out. And it releases a whole bunch of electrons that go into the electron transport chain, and that makes remember, you're trying to make usable energy in the form of ATP, and that's what's coming out here. Now, the big steps, half, cycle, electron transport chain, also happen in photosynthesis the other way around. The little bits of the steps are completely different, but photosynthesis starts with an electron transport chain, goes into a cycle, produces half molecules of glucose, and then sticks those together to make glucose. So it is kind of weird that even if you look at the little details, nothing's the same. But if you look at the big details, one looks like the reverse of the other. The same way that the basic reactions look like opposites. Speaking of which, So glucose, C6H12O6, plus oxygen, O2. The reaction releases energy that's used to make ATP. You wind up with, if you're taking chemistry, you know about balancing equations, but biologists, we often ignore it. But you wind up with 6 of these because you started with 6 carbons. And you're going to wind up with 6 of those also. So you've got a bunch of bonds to break in order to move the energy over into ATP. And the way that it gets done, each bond can produce upwards of 5 or 6 different ATPs. That energy can be used to feed through a system and produce a whole bunch of ATPs for every carbon bond that you break. So glycolysis, which literally means sugar breaking, is that okay, for people that like to understand how things work, this is for you. I'm not expecting anybody to really remember this, but I know for me, knowing the steps helps me remember the big picture too. So here's glucose. Remember a ring with a little oxygen there? ATP comes in. Here's your first ATP coming in. That little dark spot is the phosphate from here. Remember I said ATP can transfer energy, but it can also transfer phosphates onto things, and having that phosphate now stuck onto a molecule can destabilize the hell out of it. Here's glucose. There's 5 carbons in the ring and 1 carbon sticking off. Stick a phosphate on there and it vibrates and reforms, and now there's 4 in the ring, and here's 1 and here's 1 off the ring. So now there's an extra carbon up, kind of free and accessible. Here comes your second ATP in. It's going to put a phosphate there, shake the holy hell out of the molecule, and break it in half. So you go from 6 carbons to 2 of these. 1 carbon, 2 carbons, 3 carbons. Each one with a phosphate on it. These things, this picks up a phosphate just out of the juice, and it's still not stable. Each phosphate can be used, keep in mind there's 2 of these. Each phosphate can be used to make more ATPs. So you go from 2 going in to 4, 2 of these molecules, each with 2. Here's ATP being made, here's ATP being made. 2 of them, 2 of them. So 2 go in and 4 come out, and here's the half carbon molecule that's going into the next step. What else have I got here? So without showing the molecules, ATP in, 1 phosphate, 2 phosphates, 2 3-carbon molecules. I know these are really tiny. This also is usable in a bunch of stuff in our chemistry, NAD, but we don't really talk about that in this class. I do in my majors class. And then 2 ATPs get made here, 2 ATPs get made here. So 1 and 1 go in, 2 and 2 come out, and then you've got the molecule that's feeding into the next stage. So 2 are coming out here, and about 36 are coming out of the next stage in the mitochondrion, the actual aerobic stage. So this is what we get from the anaerobic stage, and about 36. It's not a straight up one-to-one correspondence, so it's sorry, oops, shoved it the wrong way. Mitochondria in here. What comes off of the sugar molecules, the half sugar molecules, as they break down, is not just energy, but it's a whole bunch of hydrogen ions. Hydrogen ions are a great way to move electrons around. You don't really need to know that, but that's how the electron transport chain works. And that is the next step. It depends on how you make your calculations. Under some conditions, you're going to get 32 out. Out of some conditions, you're going to get 36 out. So around 34 ATPs are made, 36 kind of at the end there. Actually, this should be 34 because the 36 takes into account the 2 from the first step. That's as much detail as I need you to know about this process. A couple of other things. So you're out for a nice hike. You're walking along. You look up the trail a bit, and you're out west. There's a grizzly bear. And you go, oh, I got to get a picture of it on my phone. And as you're looking at your screen, you realize that the grizzly bear is looking at you. And he's not thinking, oh, time for a selfie. He's thinking, oh, I was wondering what I was going to have for lunch. And so you take off. You're trying to get somewhere where there's going to be no grizzly bear. Maybe back to your car, maybe you get a cabin, maybe you can shimmy up a tree that a grizzly bear can't climb, but you're taking off. And you're breathing hard, and your heart is pumping, and you're circulating oxygen to your muscles. But you can't quite get enough oxygen to your muscles for what they need to do to get you away from that grizzly bear. Your muscles are the only cells in your body normally that can do this. That can, if there's not enough oxygen, okay, if we cut the oxygen off to your brain, all of your respiration stops, including the anaerobic front end part. Muscles will do the anaerobic front end part just to get some energy so that they can keep cranking. And in the case of this anaerobic production, you get lactic acid. Very similar to pyruvic acid, which is your normal product of the first stage of respiration. So say you get into your car, you get into your cabin. You got to pay off your oxygen debt. You've built up all this lactic acid. It needs to be put into your mitochondria and processed through to carbon dioxide. So you're sitting in your car, and the bear is whacking on the door, going, I just want to get a selfie with you. And you're going, and that's, you're paying off your oxygen debt. Now I said normally, your muscle cells are the only cells that can do this. During tumor development. Remember the inside of a tumor, the cells are too far away from their oxygen levels, and they've got two options. They can either turn on the gene that sends a signal out and brings blood vessels into the tumor to feed it, or they turn on the system that goes into oxygen debt, in which case they can be in a low oxygen environment and get enough energy to keep themselves alive. So this is something, the same way you don't want your cancer cells to turn on their pseudopod abilities and crawl away from the original tumor, you don't really want them to be able to do this because they'll survive better. But it's not an uncommon thing for them to do. Respiration. Breaking carbons apart in big molecules and using that energy to make ATP. Doesn't have to be sugars. It can be lipids. Remember those long strings of fatty acids? You can pick those apart, pick them apart into small bits and feed them into the mitochondria. And you can go into proteins. You can pull your proteins apart into amino acids, pull the carbon parts off the amino acids, and feed those into the system. The problem with that is you wind up with the nitrogen left over. Okay, there we go. In the form of ammonia. Ammonia is pretty reactive, which makes it pretty toxic. And animals that live in water where they can just dump it out, it's very soluble. And as long as you got lots of water around you, you can release it. And there are things out in the environment that'll use it. We don't really want ammonia circulating in our blood. We process the ammonia into urea, which is slightly less toxic, and so we can circulate it for a while before we clear it out from our kidneys. And animals, once animals were on land, one of the big problems was being able to reproduce offspring on land. Being able to get sperm to the egg cells was a big deal, and also once you could lay eggs on land that could breathe and not dry out while the embryos were developing, the embryos were also releasing these wastes inside the eggs that could kill them. So what they do is they process the ammonia into uric acid, which is totally non-toxic. So it can build up kind of off to the side in a sealed egg like a reptile or an insect or a spider, and the embryos are not poisoned by it. So these are your main what are called nitrogenous wastes, nitrogen-containing wastes that you get from breakdown of proteins. One other thing that I want to mention, calories. Remember the measurement lab? Calories is a measurement of heat, technically. Well, little c calories is a measurement of heat. Big C calories are 1,000 little c calories, because we liked the idea as Americans, but we didn't like the actual measurement size. So instead of making up our own measurement, we stole their name, capitalized it, and actually said, well, yeah, and people go, well, yeah, but it's not a calorie. Well, yeah, it's a kilocalorie, but it's our calorie. So if you see something and it goes, just sitting on your butt and you burn off these many calories, what they did was they read something that was talking in terms of little c calories and didn't realize there was a difference. And when they tell you, you think, oh, wow, that sounds too good to be true. It is too good to be true by a factor of 1,000. Okay, let me throw you one overhead here. Well, let me throw a couple of them up here. The electron transport chain in the membranes inside the mitochondria. The hydrogens pull the electrons through, and down here you're making ATPs. You're making ATPs. You're making ATPs. If you have starches, you can break those down to sugars. That feeds in, well, it depends on the sugar, really. Some sugars actually get processed into these molecules. But anyway, goes into glycolysis, fats, broken down into glycerol, which can be broken down into this. Fatty acids, which can be put right into the middle part by breaking them apart into small carbon, 2 or 3 carbon molecules. Proteins, break them down to amino acids. The amino groups, which is where the ammonia comes from, come off to the side. And what you get out of the amino acids either fits in here or fits in there, depending on how big it is. So these are all ways to feed into this process and produce ATP. Here's one with more friendly pictures on it. Amino acids feed into this. Here's nitrogen and nitrogen wastes. So this would be your ammonia, your urea, your uric acid. And you also use the amino acids to make more proteins. Starch, sugars broken into glucose, fed in. Lipids broken down into triglycerides. There's your glycerol. Phospholipids, remember, are broken down, and you can use the phosphate group, obviously. And you can make steroid hormones too. There's a whole bunch of stuff you do with the lipids. But also, we're talking about feeding it in here. Okay, next part of the chapter is about where life came from originally.